Well, today is a breakout session as we continue to investigate nutrition, and we are focusing on the obesity problem. We had a big obesity roundtable. I called it my big fat panel, where we had these two gentlemen that I'm here with today on. And I thought, let's continue to have these breakout conversations from that original kind of overview of everybody's approach to what's causing obesity in that panel that we had. So we're joined by Brad Marshall of Fire in a Bottle and Tucker Goodrich of Yelling Stop at Blogspot. So how you doing, guys?
Doing great. Happy to be here.
Good. Yeah, very good. Great to see you guys. Brad, great to see you again.
Yeah, absolutely.
So I wanted to kind of continue where we left off in the conversation. I think that was the first time you two had been on a podcast together. Is that right?
That's correct.
Yes, that is correct.
But you guys have been
kind of ironic, given we're kind of partners in crime on this whole enterprise.
Exactly. You guys have been going on blog posts and Twitter threads and everything for a long time on the topic of seed oils and linoleic acid and how this all connects to the rise of obesity and all of its different related disorders and diseases. So I thought, hey, let's just bring you two together and explore your approaches to solving this problem and understanding its cause and its solutions. I mean, that's what I'm fascinated by for a variety of reasons, but I think it just takes having conversations to get these things to cross-pollinate ideas together. And I don't think you can really do this too much in academia as easy as we can do it in the podcast world. So it makes for a little fun to do this.
Excellent. Thank you for having us here, David.
So Brad, we left off with you on our show with Georgie Dinkoff, who is known as Haidut in the Ray Peat community. And you guys had a lot of fun synergy together, didn't you?
Yeah, that was a fun conversation. And going into it, I had no idea actually how much we would agree on things. So I guess pleasantly surprised by that. Yeah, it was fun. We even talked about the traditional way of making masa dough, as I remember. That was
interesting.
It's kind of a curveball later in the show, but yeah, it's one of my favorite topics that really doesn't have a lot to do with this, but it came up. Yeah.
So Tucker, your approach, now I just want to set the table for people. So both of you guys say that seed oils are driving obesity, but you have slightly, if I may, different explanations as to how they're causing obesity, maybe slight different emphases. Is that the way to put it?
I think that's probably the best way to put it.
Yeah, I think that's correct. Absolutely.
I think we both, I think Brad and I have both learned a lot from Peter Dobromilsky. And I asked Peter a while ago, about his famous proton series, what does it all boil down to? And his response was, don't eat linoleic acid. And I think Brad and I both agree on that as a starting point. And then we can have some really interesting discussions about what happens after you don't follow that advice and you wind up having metabolic problems.
So that's the interesting thing is you guys have different explanations on how linoleic acid is causing the obesity, but then you also have kind of like different remedies for how to deal with it. So, and not that you're kind of staunch on your approaches from what I understand, but Tucker, you take more of a kind of a paleo approach to remedying the effects of linoleic acid, right? As it pertains to obesity, right?
Yeah, well, I think, and I don't know if that's something Brad and I really disagree on, but, my basic attitude is that the diet that we evolved for is going to be the one that we function best on. And that diet didn't lead to obesity up until the modern era. Now, given that we live in the modern era, then there may be tweaks and hacks and things that one needs to do to get back to a more ancestrally appropriate diet. And I think that's that is where Brad's got some it's doing some experimentations on figuring out what are hacks around that basic premise. Is that fair to say, Brad?
Yeah, I think that's fair to say. And I think probably the way that I sort of differ from that is just, the kind of observation that we can look around the world and we can see obviously lots of, and I think Peter said this in our last panel, that you can look around the world, and I know you've pointed this out too, Tucker, lots of sort of populations that are eating like white rice and don't have obesity. And now that's obviously not a very sort of Paleolithic diet approach to eating, but but we can see that that's true. And so that's kind of where I've been thinking, like when I look at it and I say, well, what, if you ask the question, what aren't obese people good at doing? The answer seems to be, oxidizing glucose and oxidizing carbohydrate, which is something that Ray Peat does say a lot. And that's why we've had him on as well. Right. And but the more that I look at it, I'm like, yeah, it seems to be that downregulated pyruvate dehydrogenase is a huge problem, and I know you've also are aware of that. We've gone around it on Twitter, and yeah,
yeah, exactly. We both agree that's a major issue, and that's what seems to drive conditions like cancer, which we're all worried about, right? The pyruvate dehydrogenase is what a lot of folks don't understand is that glycolysis takes place continually in the cell, and it produces from glucose something called pyruvate, and pyruvate dehydrogenase moves that into the mitochondria where it should be oxidized for fuel. And if you break that process, then the cell kind of reverts back to this old glycolysis formula of burning glucose into lactate, which is very inefficient and generates a lot of excess lactate, which again, ideally should be oxidized into the mitochondria. But if the mitochondria aren't working correctly, that whole process gets foobarred. And that seems to be one of the fundamental aspects of cancer cells. So yes, avoiding that process is a good thing.
So my biggest thing is I'm interested in what the actual driver of obesity is from the seed oils, but I'm more interested in helping my audience, because I talk to a broadcast audience that's not looking up seed oils or nutrition or paleo or Ray Peat. These are people that are just everyday folks driving in their car and flipping through the dial, and they hear something different. What is this seed oil thing? So I'm trying to think, okay, what is the best path of healing from obesity and all of its related effects? Once you identify that the seed oils did cause the problem. So that's why I want to know the answers. I'm like a hound. I want to know what is the truth here. Is it just multiple paths? You can do the high sugar thing. You can do keto. It's all washes out as, whatever. Or I don't like that. I want to find the truth. I want to find the best solution. And most people will say, well, it's all up to your body, do this. That's like the easy way out, but maybe that's true, but I want to know. I want to know if that's, so that's kind of where I see you two helping me understand this better is, Tucker, your approach is not to take more novel interventions, but rather to, approach a keto, low carb, low linoleic acid, right? That's your approach. That's what you do. Hashtag LC L6, right?
Yeah. And I think, based on our evolutionary history, that's the diet that we evolved to eat. And, this is basic systems engineering 101. If you have a complex system that, I come from an IT background, and we were dealing with systems with millions and millions of lines of code that we didn't have access to. So we had no way of knowing what was going on inside the system often, which is very similar to where we are with biology and human biology. It's a very different thing to say that you've mapped the whole genome and then going on to say, but we understand what's happening, and we clearly don't understand what's happening. So whenever you have a complicated system that is messed up, the first step should always be take it back to the known good input state, right? Because typically bad inputs cause bad outputs, right? It's just like, this isn't rocket science. If you've got sugar in your gas tank, your engine's not going to work right. First thing to do is clean out the gas tank, get rid of the sugar, and just go back to straight gasoline. And then you're probably going to be in a really good place and not have to worry about anything else. Like, what are the mechanics of your car engine, right? Because it's an input problem. It's not a problem with the system. And I think that that's, so going back to our known good input state, right? Our ancestral diet is the first thing that anybody should always try.
Brad, why not just do that?
Well, so this is actually good timing because I just, I had coffee with my mother yesterday, and she was diagnosed, I want to say 3 years ago, with sort of early stage type 2 diabetes. And she in the past has done a ketogenic diet many times. And so that was her, that was her natural thing that she did. And she was able to lose a significant amount of weight. And I think her blood sugar came down. But as I've seen a lot of times with people who go on a ketogenic diet, she has not been able to maintain the diet, and so now she's regained a lot of the weight, and she's kind of struggling with, do I want to go back to that? And I think for a lot of people, being on a ketogenic diet is difficult. And so that's kind of why, that's one of the reasons that I'm looking for, different approaches that can also work. Like, so I guess one way that I think about it is that, okay, so we know that we have this problem that we can't, that obese people are not good at oxidizing glucose, right? And
Brad, before you get into
Sure.
that, I had dinner with Eric Westman at KetoCon up in Connecticut a couple of years ago. Eric Westman is a famed low carb doctor,
right?
And I asked him, and he's got thousands of patients, so a really interesting guy to ask this question. And I asked him, does this ever not work? And his answer was, no, it always works, but some people can't follow the diet. And for those people, we need to do something else. So
Right.
yeah, I totally agree with, for some people, for whatever reason, and it just, if you can't, it's like any sort of weight loss diet. If you can't stick with it, it's not a permanent fix. You can't go on keto for a week and then you're cured for life. You got to stick with it.
Right. And so I think that's exactly right. And so I've been, I've moved on to thinking more about ways, how do we actually, if I'm obese, how do I again become a person who can basically oxidize glucose efficiently again, right? Because I feel like that really is, I feel like keto is something of a dodge, right? Because you're not oxidizing glucose well, and you can go keto and you can lose the weight, but
you're not fixing the underlying problem.
You're still in glucose sparing mode, right? You're still not really metabolically flexible in the sense that you can go back to eating carbohydrates and you're not, right? It's going to stop working.
Right. I totally agree with that.
Yeah. So you haven't really fixed the problem. And so that's kind of what I'm trying to figure out ways to do. And obviously that's, if that were easy,
we wouldn't be having this conversation.
Wouldn't be having that conversation, right? Yeah, exactly. So, but nonetheless, I think we do there are a lot of ideas out there. And one of the things, and so this is right. It's okay. So now we're looping back to vegetable oil and why linoleic acid and what the problem is. There's a lot, I guess we have some agreement that the getting the pyruvate dehydrogenase complex working again would be the key step in doing that, right? I mean, I think, I don't know, would you agree with that, Tucker? Yeah. And
it's definitely a key step. Absolutely.
Yeah. And so then it's like, okay, so we know that that is inhibited by some of the things that build up in the mitochondria, like NADH and acetyl-CoA directly, but also, and this is where I would defer to your knowledge, it can also be inhibited by the oxidized linoleic acid products. So these are when the vegetable oil oxidizes, it can damage proteins. And
one of the proteins that gets damaged is pyruvate dehydrogenase. And another one is, complex 5 of the electron transport chain, so it can't produce as much ATP as it should.
Right.
So you've got these multiple, and I think this is, back to your introduction, David, there's a lot going on. It's not like there's one thing that's happening in obesity. And so I think there's a lot of conversation to be had over what the different pathways are and what we can do to, what we can do to tweak things. Because a lot of this protein damage stuff, for instance, it's not exactly clear how to fix it aside from just waiting. So, I mean, it's, if you've broken something, and the example I often use is cancer, is we have a lot of reason to think that linoleic acid is involved in various cancers, not all cancers, but some cancers. And once you get cancer, can you cure it by going on a low linoleic acid diet? And I don't think there's any evidence for that, because then you've got something that's broken and you've got to figure out, okay, how do I address this problem? I can't just go back to the default diet because something's broken, and it's going to continue to be broken until I can address that particular problem. So I think that's kind of where we are here with obesity, is we know that things get broken in obesity, and how do we try and fix those things? Or how do we at least try and accelerate the healing of those things?
Right. Yeah. And I'm a person that I've stopped. I have avoided seed oils for the most part as an adult, although there's, times when I've been better about it than others, but very strictly for the past 5 years, I've avoided seed oils. But it doesn't, that doesn't automatically make me a lean person unless I'm very strict on a ketogenic diet, etc. But I would like to figure out ways to right. Yeah. I'm trying to get better at burning carbohydrates and being able to eat a mixed fuel diet. So we'll keep seeing how that goes.
But Tucker, how come, how do you have any idea about how people can burn carbohydrate healthily, at a higher carb diet?
Well, I mean, it's useful to remember humans do have a genetic adaptation that we can track to a high carbohydrate diet. And this is the enzyme that we use to break down starch into glucose, right? And this is something that we actually have in common with dogs who, we domesticated and generally feed on people food, and they have the same amylase adaptation. So people or dogs who've been living on a high starch diet for a long time have high levels of the copy of this gene that makes amylase, and people who don't, right? So it's an adaptation to a high starch diet. Now, does that mean, and that much is clear that exists, but I've never really heard a good explanation about what the adaptation is actually doing. Is it protecting you from negative effects of too much glucose, or is it a positive adaptation in that it's allowing you to extract more energy from a high glucose diet? You could argue either way, but one can make the argument that if you've only got 2 copies of the amylase gene, that you're well adapted to a low carbohydrate diet. And maybe one of the problems that adaptation was designed to protect against was, inappropriate weight gain, because in a traditional human lifestyle where, exercise and running around is a major part of your survival, that being overweight is not productive and leads to injury. And, we have a huge study in the Air Force showing, the military has a huge problem now with running injuries. Well, running is one of those basic human adaptations, right? It's what we evolved to do. And yet that's the biggest cause of injury in the military now. And when we look at what's actually causing that injury, it maps almost perfectly to being overweight and trying to run. It's not a being overweight and trying to run is not a survival trait. Okay. So yeah, it could be as simple as, we don't have a genetic adaptation for that, right? I mean, we know with Europeans, we're all of European ancestry, obviously, that there were two populations in Europe. There was the European hunter-gatherer population, and they were largely wiped out and overridden by immigrants from the Middle East who were much more adapted to a high starch diet. The obvious example of that is the Romans. Right. So maybe that's just it. Maybe you've got a different genetic pattern that makes you less well adapted to a high carbohydrate diet. In which case you may not, there may be hacks that you can do, but you may be up against a genetic background that's going to make it really difficult to adapt to it in your lifetime.
Right. And that's possible. And there's also, but we also know there's underlying, we do also have underlying metabolic changes that I think are, can be kicked off by, by eating seed oils in the first place. And then,
yes,
right. And they may not necessarily turn off just because we've stopped eating seed oils.
yes.
And really what I mean is kind of the de novo lipogenesis system. There are all these enzymes and, obviously the SCD1 is the most famous, and I talk about it on my blog a lot, but, what happens is you can see that if you look at the body fat of starch eating cultures, it's very saturated. And what happens is over time, the seed oils kind of kick off this process of upregulating these de novo lipogenesis genes, and they make your fat more unsaturated. I mean, that's ultimately what they do. And so then when you look at obese people, they have lots, and the fat gets less and less and less saturated. And even though you take away the polyunsaturated fats that you get from the diet, your body fat is no longer made of the saturated fats. It's made largely of monounsaturated fats. And that seems to be sort of a self-reinforcing cycle. And so that's a tricky one to understand how to get back out of, because, you have like all of these thousands and thousands and thousands of extra stored calories that's in this form that's maybe not helping you. So how do you,
yeah, what do you do about that? Right. And
right.
And we don't have an answer to that at the moment. I mean, there are some logical things that you can do. But the estimate for how long it takes to fix that is based on how long it takes to cause that problem. So the short answer is, if you go on a low seed oil diet, how long will it take to reset your adipose tissue to an evolutionary fatty acid balance? And we don't really know that.
Yeah.
And it may take, different, we don't know what the interventions are that actually work. None of that's been tested. Because to test it, you'd have to admit that it's a problem. And
Tucker, have you tested yours to see if you're
my
highly
adipose tissue?
Yeah.
No, I wouldn't expect it to be as highly saturated as what Brad is describing, because I'm not eating a lot of starch and I'm eating a lot of animal fat. So I should have higher levels of, oleic acid and probably long chain omega-3 and omega-6 fats than somebody who's eating just a high starch diet, because starch in lipogenesis gets converted to palmitate, which is a fatty acid, and that gets converted to some oleic acid and some stearic acid. So if you're eating a high starch diet, that would be what you'd expect to see your fatty acid or your adipose tissue composition be. And I honestly haven't looked at that, Brad. Have you?
I mean, the only way that I know to do it, there's an Omega Quant, the company's called Omega Quant, and they have a test that you can look at the red cell membranes, red blood cell membranes.
Blood cell, right.
Right, but it's blood cells and it's membranes, both of which are different than your adipose tissue, but there is definite correlation between what's in your adipose and what's in the blood cell. But of course, it's not an easy one-to-one correlation. So it's
also your blood changes in the short term and your adipose tissue only changes in the long term.
Right. Yes, exactly. And so it's imperfect, but it's the best thing that I know about.
Right.
At least gives you an idea. Yeah. And those other, And the other thing you can do is test your, like you were saying about cells creating lactate. Well, I've been thinking more recently about, what we could do. And one thing you can do is you can look at the lactate to pyruvate ratio. in your bloodstream, which are both tests that you can get at a normal, like at a normal blood testing lab. That gives you, well, it gives you really, it tells you your redox balance, whether or not you're in a more of an oxidized state or reduced state. Just complicated thing to say, I know. There's an easy way to.
I knew we were going to get there, though.
Well,
Yeah,
I mean,
I was just about to ask that.
that's kind of what that's what I'd like to know, but I don't actually know that. I've never actually gotten that test done. You have to go to like a LabCorp or something. And, Dave and Shebog Huggins have a service on your lab that you can, in theory, get these tests done, except not in New York State where I live. So it's frustrating.
Right. Now, so explain to us, well, pyruvate and lactate are both molecules that are produced from glucose. What would you want to see that ratio? Would you want to see it biased more towards the lactate or the pyruvate?
I would want to see it biased more towards the pyruvate.
Pyruvate, okay.
Right. And so, like you said, cancer cells are exporting lactate
Right.
because they build up NADH, and the way that cells kind of, so, the NAD plus to NADH ratio is very important. You need NAD plus to run like the Krebs cycle and to do basic metabolism. And a lot of things end up low in NAD plus, and NAD plus goes down as you age. And so, but what happens is cells can actually import pyruvate out of the bloodstream, and they can, lactate dehydrogenase will.
It goes up as you age in an industrial diet context.
Well,
and I'm guessing it's never been tested in an ancestral diet.
I'm sure you're probably correct about that.
Right. Okay.
But right. But so one thing that happens is pyruvate is imported into the cell. You convert it to lactate and you re-export it. And so what happens is that lactate and pyruvate in the bloodstream is pretty tightly correlated to the NADH and NAD plus ratio in your cytoplasm. And that seems to be very accurate in real time. So that sort of tells you if you're all of the desaturase enzymes like D6D and SCD1 run off of NADH. And so if you have high levels of NADH, you're likely to have high enzyme levels of the enzymes involved in de novo lipogenesis. So it's one test that we can do that kind of tells us where we're at.
Right.
I don't know. There's not a lot of great tests out there, unfortunately.
Well, there's a scientist out in Colorado, Inigo San Milan, who's been doing a lot of work on lactate in cancer and tying it into the mitochondria. And it seems to be a pretty crucial. I haven't honestly been paying super close attention to his research, but yeah, it does seem to be a really fundamental health sign. If that system is off, then it indicates something may be dramatically wrong.
Yeah, and I think that on average, and if you look at obese people, they have a high lactate to pyruvate ratio in obesity and diabetes. It seems to be the general rule, and so that's another thing that we could test, and perhaps there are ways, and that's, I guess, an area that I'm very interested in is how does one get more NAD plus back, and because low NAD plus is very much associated with aging, obesity, diabetes, and it's a very crucial cofactor for like I say, our whole metabolism. Basically, you need NAD plus, and so how do we restore that naturally, without, I mean, you can take NAD plus boosters, but I think that by thinking about, I mean, sure, the last article I wrote was about the differences in how a fat called stearic acid is transported and digested and oxidized compared to how things like linoleic acid, they behave very, very, right. They're both fats. And so people say, well, fat is fat, but in fact, once you consume them, they act very, very differently in the body. And I made the analogy that stearic acid is more like it's almost like fiber of the fat world. It's very slowly absorbed. It's very slowly transported around. It moves into the mitochondria very slowly. And it's kind of like this slow running diesel engine. Whereas if you eat vegetable oils, linoleic acid, it's very showy. You know, you're driving a Porsche or a Lamborghini, but those cars can have problems, especially, you know, if you're going 200 miles an hour, you can get off the road pretty quick. And so that kind of, you know, there's very fundamental differences in how different fats are dealt with by the body, all the way from, the whole level, all the way down from starting with how quickly they're absorbed, starting with how quickly they're burned, and then all the way down to how much a reactive oxygen species they make when we burn them into the mitochondria, which is, of course, what Peter Dobromilski talks about a lot. And so it is, well, as you know, this is multifaceted, but it's very interesting. And we see if you feed mice, for instance, so this is what got me interested in this topic in the first place, is if you feed mice a diet that's basically combines stearic acid and starch, they have like no abdominal fat. And if you feed them a diet of starch and vegetable oil, the same number of calories, they have significantly more abdominal fat. And so what causes that? And so, that's one of the things I've been thinking a lot about. I think that in general, and this is another, David, we've talked to Ray Peat, and this is an interesting thing, the idea that actually slowing the rate down at which we are burning fat actually helps the mitochondria to kind of burn cleaner, and it prevents the rapid buildup of NADH, and it kind of, just keeps everything burning cleanly and keeps us getting enough oxygen basically into the system. It's sort of similar to what people will argue for dietary fiber, right? It slows down the glucose absorption. It gets us going at a better rate. And I think that the saturated fats perhaps play that role in, in actually burning fat. So I don't know if.
Tucker, what do you think of the idea of stearic acid as a kind of fiber of fats, as a picture?
I don't know. I haven't really thought about that quite that way before, honestly. I mean, stearic acid, for the audience, is something that we likely would have eaten a lot of in our evolutionary history. It's a, common fat that comes in ruminants, so we should be pretty well adapted to it. But it's interesting that it's one of the ones that we seem to have it's kind of got a complex absorption pattern, and there's some evidence that we don't absorb it. And it might be that the body's saying, for instance, iron, if you have enough iron in your body, you absorb less of it. So perhaps because that fat is something that we're so well adapted to that, and was such a common part of our diet, that we kind of, don't absorb it, and it slows things down. So yeah, I think there's definitely, it's an interesting argument to make.
Slow and steady wins the race is your idea, right, Brad?
Yeah, I mean, I think so. And like I say, that is an idea that I absolutely, one thing that got me going down that path was something that Ray Peat said in our larger obesity panel. I think what he said was, polyunsaturated fats are oxidized too quickly. And that's the problem. And that prevents us from being able to oxidize glucose. And that got me thinking about why would polyunsaturated fats be absorbed too quickly? And it turns out there's a lot of evidence to suggest that that is indeed the case, and that the saturated fats slow things down. And it's a slow and steady wins the race kind of argument. Absolutely.
Tucker, what do you think of that overall idea that it seems like Brad and Ray Peat are in large agreement about, that reductive stress is what's causing the obesity problem with linoleic acid?
Well, I don't agree that that's what's causing the obesity problem with linoleic acid. And I think it may certainly be part of the process that's going on. I mean, Brad and I went back and forth on Twitter the other day with our rodent knockout models. So genetically modified rodents that don't have a this or that gene, right? As a test of, is this or that process driving obesity? So if you take rodents and you knock out their endocannabinoid receptor, which is, the endocannabinoids are a signaling molecule, class of signaling molecules. If you knock that out, they don't get obese, right? And that, we know from later research, that happens in the gut before the food's even absorbed, right? So from that standpoint, that's a controlling element. Now and then Brad, I think you were pointing out that if we knock out the SCD enzyme, am I correct in that? Or am I misremembering?
No, that's right.
That they're also protected from obesity, right? So we've got, first step, the food hits the gut lining before it's even absorbed into your body. We have this signaling pathway that kicks off. If you prevent that, you prevent obesity. Then if you don't do that and it's absorbed into the body and you block this other step, then they're also protected from obesity. So again, back to, what we were saying at the beginning, there are these multiple pathways that are happening, and often they're happening in parallel. So no, I don't think that's what's causing it. I think that may be in the causal chain somewhere down the line, that, okay, so this happens and this happens and this happens. And then if SCD doesn't get triggered, do they not get obese? Yeah, that's, that's what the studies that Brad was citing show.
So give us an analogy. Give us like an economics analysis or analogy or something like that to help this, what you're saying, Tucker. It's like you're saying it's not the cause, but it's a downstream part of it. Like if you're looking at a recession, that this is not the cause, it's that, or give us an.
I prefer to go with the mechanical analogies. Okay. I mean, it's like the difference between your fuel pump being broken and your gas tank being empty, right? They have the same effect, but there's two different causal nexi, right? There's two different things that are happening. I mean, if your gas tank is empty, obviously the engine's not going to run, but you can have a full gas tank and the engine still won't run because your gas pump's not working and you're not delivering fuel into the engine. So, right. And I mean, it's not fair to say if your fuel pump's broken that the problem is you're out of gas, because you're really not out of gas, right? You've got a broken fuel pump.
And I tend to think that these things are not mutually exclusive, that, like Tucker says, there's a lot of things running in parallel. And I think they're all little parts of the answer. I'm sure that I don't disagree with Tucker. I think the endocannabinoid system is very important in all of this. I think that, and Tucker, explain how the endocannabinoid system relates back to vegetable oils.
Well, so you eat linoleic acid. Let's just start at the beginning. You eat linoleic acid in your gut. It's in your intestines. It's converted to a fat called arachidonic acid, which is a longer chain omega-6 fat. That fat is converted into endocannabinoids, right? So this is a pretty straightforward pathway. The two best studied endocannabinoids, anandamide and 2-AG, both, they work together to control hyperphagia. What's hyperphagia? It's overeating, right? So it is literally your body's. Now, what's another way that you can upregulate these endocannabinoids? Don't eat. When you don't eat, your body upregulates production of these endocannabinoids because they tell your brain that you're hungry, okay? So basically, if you eat excess linoleic acid, you generate a signal that tells your brain that you don't have enough fuel. Oops, and it makes you eat more, right? And we know that this is true, and this has been demonstrated so many times in mice, it's not even worth going through the studies. But we know that it works in humans. because another endocannabinoid is one that we've all heard of, THC, which is in marijuana. THC gives you the munchies, and it's used as a drug, an FDA-approved drug called dronabinol. And this is prescribed to people with cancer and people with AIDS who don't want to eat because it makes them eat, right? So this is as clear as it gets. And we have another used to be FDA-approved drug called rimonabant, which blocks this pathway. And if you do that in rodents or in humans, then you lose weight, right? But what's interesting is it has all sorts of other effects. So rimonabant also causes you to stop being insulin resistant, right? So effectively what the endocannabinoid system is this regulatory framework around your energy metabolism. And, to what Brad's been arguing, SCD1 is part of that, the plumbing of the energy system. And it may be that also seems to be regulated by endocannabinoids. And it may be, as Brad is suggesting, that if you can tweak that element of the system, that you're altering how an important part of the system works that will then go on to control your obesity or leanness phenotype, as is evidenced by the SCD1 knockout rodent models, right? Because we're talking about, people go back and forth about rodent models and human models. This is an area where the correlation between the two of them is pretty good. Insulin metabolism, endocannabinoid metabolism is something that's pretty common between, the animal models that we use in humans. So it's one of these areas where, we can, as has been demonstrated by the fact that THC and rimonabant both have the same effect in rodents as they have in humans. We know that these systems are pretty similar, although not identical. Is that fair to say, Brad?
Yeah, I think so. And just but then again, to explain how sort of intertwined and overlapping all of these systems are. So the kind of rate limiting enzyme in converting linoleic acid to arachidonic acid, where it can then become the endocannabinoids, is an enzyme called D6D. Delta 6 desaturase is really the name, but usually you just see it as D6D. And that D6D has been shown to be, they do what it's called a not good at speaking, Mendelian randomization study. And this is basically the type of study in humans that is the next best thing to doing, a controlled study where
claimed by the people who use it, although
claimed by people who use it. Yeah,
I will dispute that, but that's the topic for another podcast. But go ahead and say what you want to say about it.
Sure,
it can be. It's definitely can be a useful tool, no question about that.
Right, and there are a lot of but there's a lot of evidence that the activity level of this enzyme D6D is, like I say, it's converting linoleic acid to endocannabinoids. That's what it does, and that it is causally linked to diabetes progression, etc., in humans. And the thing is, the activity level of that enzyme is driven by NADH levels. NADH is the cofactor that goes into the reaction of delta 6 desaturase to convert linoleic acid to send it down the pathway to becoming an endocannabinoid. So if you're in reductive stress, if your cell has a lot of buildup of NADH, you're going to have high activity of D6D, and that's going to add to the endocannabinoid problem, right? And
it's causing you to overproduce endocannabinoids.
It's causing, right, the reductive stress itself is causing you to overproduce the endocannabinoids, which is the thing that's giving you the munchies, right? And so they, like you say, it is obviously it's complicated. We have all these systems running in parallel, but they really are intertwined and multifaceted, and it's hard to it's hard to always tease out what the where do you start, what's the cause.
Well, yeah, where do you stick your lever if you want to move the system,
right? Yeah, exactly. But that's one of the reasons why I think
so.
I've been thinking, obviously I've been thinking, that's what I've been writing about my blog, is reductive stress, and like that's one of the reasons that I'm interested in it. Yes.
So can you explain to me and to David and our audience, because I'm not as well versed in this as you have. You've been doing a lot of research on this, and I've not followed up on it. And thank you for sending me a whole bunch of papers on Twitter the other day that I haven't gotten through yet. So explain to us what can this is another system that's getting dysregulated in this process. So how does it get dysregulated?
So there's several ways. One is, so one of the things that obviously going back to Peter Dobromilsky, and this is the protons thread that Tucker mentioned, is different fats, when they're burned in the mitochondria, essentially produce different amounts of something called reactive oxygen species. Those reactive oxygen species play multiple roles, but one of the things that they're doing is they're increasing the amount of oxygen essentially burned in the system. And the way that we get rid of reactive oxygen species is with something called glutathione. And this enzyme, so glutathione is the body's main antioxidant. I won't say them, it's one of the main bodies.
Oh, I think it's fair to say it's the main antioxidant. I mean, it's one of the few
after superoxide dismutase. Then
okay, but I mean, it's one of the few where if you have a genetic knockout of it, it's fatal in the fetal stage.
Right, absolutely. No, it's a very important antioxidant.
Yeah.
There's no question about that. And so then this thing called glutathione reductase comes along, and it actually uses a molecule of NADPH to get rid of each hydrogen peroxide molecule that's being generated in the mitochondria. And then there's another enzyme called NNT, and it basically switches the NADPH to be an NAD+, and we need the NAD+ to run the
next step in the cycle,
the TCA cycle, and
right.
that's how we burn our foods. And so what happens is the vegetable oils, they don't have I'm not going to go through the full mechanism, but they don't, when they're being burned and assuming they're not oxidized and assuming they're burning cleanly, all of which we could argue about, I'm sure, they simply don't produce as much of these reactive oxygen species, and therefore they're not giving you back as much NAD+ for every molecule of fat that's being burned. And I think that kind of slows down the system, as well as the fact that the vegetable oils are simply being sort of shuttled into the mitochondria at a rate that is probably in excess of what we want. If we were eating an ancestral diet, like you say, of animal fat, we would have fat that enters the mitochondria slowly and burns cleanly. With vegetable oils, the reasons are complicated, but it enters the mitochondria quickly and it burns what I've argued is kind of dirty, because it's not stimulating this system that's regenerating the NAD+. And so that is why the vegetable oil, I would argue, is giving us this buildup of NADH in the first place. And then that is triggering delta 6D or D6D and SCD1. And those things are, just continuing to build the problem by triggering the endocannabinoid system, etc. And so that's kind of my take on how linoleic acid is affecting things in the mitochondria and why it's causing this buildup of NADH in the first place, or lack of NAD+, however you want to think about it. But
interesting. Okay, so yeah, it's just another basic dysregulation. It's not producing the reactive oxygen species signal that it should be, that the other fats are. And so the next step in this system isn't happening in the way it should be. And you wind up with this dysfunction of too much reductive stress, too much of this reduced molecule, meaning, contrary. And I mean, just for the audience, reductive stress is basically it's kind of the inverse of oxidative stress, essentially what it's saying. And I guess the best example of this in people is that every time, oxidative stress is and is a problem and certainly can be a problem. And they've tried repeatedly to address oxidative stress by giving antioxidants. And essentially, every time that they do that, the antioxidants make health worse because they're kicking you over from what ought to be a finely balanced system by the body into a system with too much antioxidants, which is pretty much the definition of an overly reduced environment, which is reductive stress. Is that a fair way to?
Yeah. I would absolutely agree with that. And the only other thing I would say is that, well, a couple of things. One is that I think that ultimately the source of what I really think of reductive stress as being, let's take a step back, is essentially a buildup of too much energy in the mitochondria. The mitochondria NADH is used to make ATP, and acetyl-CoA is all of our foods are made into acetyl-CoA, glucose, fat, ethanol, essentially all of them, even some amino acids, are made into acetyl-CoA to get burned in the mitochondria, and that acetyl-CoA is making a bunch of NADH. And if you have too much acetyl-CoA and you have too much NADH all in the mitochondria, that's reductive stress, and it looks like energy excess, right? Right. And so then you have to ask, well, why is there energy excess specifically in the mitochondria? How did we get there, right? And so I had another point that I've now forgotten. I'll say one more point about the previously I was talking about NNT, and it's this crucial enzyme in the mitochondria that's using these reactive oxygen species to regenerate NAD+. And interestingly, and I always forget, Tucker, I'm sure you've seen these mice models. The main rats are like, Peter calls them black 6, but it's this string of letters that I can never remember. It's like N56.
Yeah. CBJ, BL/6.
Right.
Something or other. Yeah.
Right. And so there's different strains. The strain that ends in /6J is from Jackson Labs.
Yep.
And people use them in a lot of experience because they're really good because they're really easy to make fat. And it turns out they have a deletion of this NNT gene. And so when you study those mice, they have a metabolic rate that's maybe 10 to 15% lower than normal mice because they lack this enzyme that allows them to regenerate NAD+.
And they're of course used in these studies because they're obesity prone, because scientists like to know when they're doing this experiment that they're going to get what they want, which is a fat, sick mouse.
Right, exactly. And so you see how those mitochondrial dynamics are important with these mice, and that's actually how we know as much as we do about the NNT gene. But yeah, but
I didn't realize that.
So it's obviously it's very complicated how when we're talking about mitochondrial dynamics and how does linoleic acid work different from palmitic acid. I mean, there's a lot of things happening. This is very complicated. It's a very low level science, but it's surprising how much we do know.
Right. And it gets into, another parallel pathway that's going on at the same time. One of the linoleic acid oxidative products is this molecule called HNE for hydroxynonenal. And one of the many things that HNE breaks is the Randall cycle. And the Randall cycle is basically what partitions the fuels that the mitochondria is using between glucose and fat. And what it does is predispose the cell towards fat storage. So you've got, it sounds like a double whammy going on. If you're on a high linoleic acid diet, you're going to be producing excess acetyl-CoA, and then you've got HNE damaging the Randall cycle, producing excess malonyl-CoA, which is what acetyl-CoA gets converted into before it gets converted into fat for storage. So you're biasing through two separate pathways the system towards producing excess fat. And, that and, the endocannabinoids making you overeat, particularly omega-6 fats, and there's your obesity epidemic.
Right. And no, I 100% agree with you. And you see the effects of the linoleic acid on all different levels, as you just described. And so anyway, that's one of the reasons that I like about when we think about our different models of obesity, one of them is mammalian torpor. So mammals, if you feed them, we have hibernating animals, right? And so it turns out that these hibernating squirrels, one of the necessary things to get them to be able to sort of turn their metabolisms off is you have to feed them enough linoleic acid, or they fail to hibernate if they don't get enough polyunsaturated fat. And then when I look at a system that's doing exactly what you just described, we have these problems at very down low level to mitochondria, and it's trickling its way up to the endocannabinoid system, and de novo lipogenesis is happening. Maybe the system,
which would be beneficial if you're trying not to use energy, which is the case in hibernation.
Exactly. And so it's like people are thinking of obesity as pathology, but maybe the system is working exactly as it's designed.
Except that, anything, having your brake pedal stuck down is pathological when you want to go. It's a good thing if you're trying to stop, but
right. But I just, people are like, why, when you think about how all the different levels that linoleic acid is breaking thing, that's when I think, oh, the system is obviously designed this way, because what we're seeing these positive feedback loops that are all reinforcing each other. And you're like, okay, well, obviously that's how it's supposed to work on, if you're a hibernating squirrel. Obviously we don't want it to work that way. So don't eat linoleic acid, as Peter said.
Right. Exactly.
So Tucker, do you believe that we're in a state of torpor if you're eating too much linoleic acid?
Yeah, that's it's really interesting. I think it was Dr. Lustig, the anti-sugar guy. He talked about an experiment he did with human kids who had a tumor that caused them to overproduce insulin, and they were grossly overweight, and they were very they had torpor, right? It's the only way to describe it. They weren't very active, and they were hugely overweight. And so one of the things that he did was he gave them this very nasty drug that you wouldn't give to anybody else, because it has some pretty bad side effects, but it blocks their insulin production. And the first thing that happened is they became more active before they started to lose weight. The first thing that happened was they immediately lost that torpor aspect, if you want to call it that way, it that.
So Brad, knowing what you know and how much it parallels with Ray Peat's work, how come you're not exploring his solutions, about, just going hard into a high sugar, low fat, moderate to low protein? Why not do that? Why do all these things like sterculia oil like you're doing and succinate and all that?
I actually have. So one of the things I've been doing is I have actually been reintroducing a lot of carbohydrate. Most, for whatever reason. I know Ray Peat likes sugar, and I don't quite understand why he specifically likes sugar as opposed to starch. But
it's the fructose.
Yeah. And
it bypasses the insulin. Is that what it is? Because it goes to the liver?
Yeah. There are some studies, and Peter shared a study, may not have been Peter, it may have been George Henderson, showing that there are positive effects of fructose. Fructose isn't a bad guy, right? We evolved from frugivorous animals that ate a lot of fruit. And, our sperm runs on fructose, for instance. So 2 cheers for fructose, 3 cheers for fructose, which isn't saying that, like anything else in the body, if you have too much of it, it's not going to be a bad thing. But I think demonization of fructose goes too far, which isn't to say that you want to make it 100% of your diet. But there are some positive aspects to fructose, and, so it's an interesting argument. Fructose, sugar absent a high seed oil diet appears to be a lot less harmful. So let's start there with something that Pete and all of us agree on, that, the starting point should be reducing your linoleic acid diet content in your diet. And then once you do that, then you can get into, okay, does fructose go from being harmful, which it is on a high seed oil background, to beneficial on a low seed oil background? And that's a really interesting discussion to have.
Yeah, very much so.
So Brad, you're integrating Ray Peat type stuff into your other experiments?
Yeah, I mean, Ray Peat has also talked a lot about reductive stress, and so that's kind of like where I'm sort of at, is how do we get the mitochondria to run again? And so
going back to Tucker's metaphor, there's sugar in the gas tank. Ray Peat would say you need to overload it with more to get it to run.
Yeah, more sugar, right, to get it to
well
run.
that sounded like a good analogy when I said it, but it may not be the best one for this particular case.
Yeah. So I'm trying to get I'm basically trying to do things that are gonna get hopefully be able to burn glucose again. I mean, that's my goal, right? And I don't.
How do you know when you're there? How will you know when you're there?
Well, so one thing is just monitoring blood glucose, and that can be first thing in the morning, or even just sometimes I will literally, I don't have a continuous glucose monitor, but sometimes I will literally eat. I kind of have a test meal that I make. It's basically white rice with a little bit of sauteed vegetables and some lean meat, and then I will sit there and prick my finger every 15 minutes and take a blood glucose to make a curve to see how that's changing over time.
So you're measuring it based on glucose disposal, on the assumption that if you're processing glucose correctly, you're going to have a low curve.
Right,
like
but the problem, right, but the problem with that is I don't know if I'm oxidizing it or if I'm just making it into fat. Right, that's the hard thing to really know.
You need to look at RQ also.
Yeah, you need to look at respiratory quotient, and that is a much more expensive proposition.
$300,000 metabolic bench to do that.
Right, exactly. And so that's what I really want to know. And there are some, I mean, lumen is a relatively inexpensive thing, but I haven't tried it yet. I don't know. Right. I think it's supposed to, I can't figure out from their website what it's actually measuring or what it actually tells you, or if it's just like,
I think it is supposed to measure RQ,
runs you through their proprietary software and gives you like diet recommendations. Like, I don't want that. I just want to know.
Yeah, and so
quotient is.
Just to step back for a second for the audience, RQ, respiratory quotient, looks at the ratio of oxygen inhaled to carbon dioxide exhaled, and that tells you what fuels you're burning, right? Ranging from, if you're burning nothing but glucose, you're at a 1. And if you're burning nothing but fat, you're at 0.7, right? And there are a couple of little caveats in there around protein oxidation and ketones and stuff like that. But basically it's 0.7 to 1. So if you're burning 0.7, you're on a perfect fat, 100% fat. 0.1, you're at 0% fat. So ideally what you want to see when you're, when you take a glucose load is you want to see that go up, because one of the things you should be doing with excess glucose is, using your metabolism to get rid of it. And then, there's some other things that happen too, like converting it to fat. And I mean, RQ, I think, is one of the most interesting metrics that we have, because it's one of the few things that actually predicts fat gain. And it's not surprising, because basically if you're running at 1 all the time, that tells you that you're never burning fat. And we know that your metabolism is converting excess glucose into fat. So obviously, if you're constantly making fat and you're never burning it, guess what? You're going to get fat.
Yeah.
So,
it seems like the Ray Peat model is basically to get you to start eating like you did when you were in first grade, like lots of orange juice, milk, jelly babies, Mexican Coke, and I don't mean that in a bad way. I mean, because that's when your thyroid's strong, right? That's the narrative I'm hearing from the Ray Peat community, is like, you basically, everybody starts off with their thyroid basically running the show. And then because of all the damages that we do, like linoleic acid and stuff, we become like a cortisol driven body system. I'm not quite sure the narrative.
Well, yeah,
but
cortisol is a steroid hormone that your body produces, and among other things, what it does is cause your body to convert protein into glucose, right?
So they're saying like basically they want to revivify the thyroid with all the big high sugar interventions and low protein and eating gelatin proteins to counteract muscle meat proteins, which is something I don't hear keto people or carnivore people talk about a lot. I know they do somewhat, but
sorry, what was that point?
Eating lots of gelatin to balance the proteins in muscle meat.
Oh, right. Yeah, that's one of his points. And
all these things are designed to like revivify the thyroid and to drop cortisol, because apparently we all start off like little kids eating in a way that is very thyroid friendly. And then because of all the linoleic acid and stuff, we destroy that. And then we run on cortisol and we run our organs into the ground with it, with a high stress.
You know, the gelatin thing, that gets us right back to an ancestral diet, because typically now what we eat is muscle meat. We cut all the gristle off, right? The gristle is the tendons and the hard chewy bits. Well, that's what you make gelatin out of. So basically, because we're not eating that stuff anymore. I mean, if you go to Japan, for instance, tendon is considered a delicacy. Oh, yeah. And if you go to a Mexican delicatessen, they're going to have lots of tripe and pig's feet and chicken feet. That, you know, is what we make gelatin from. So it's back to, yet again, trying to fix our divergence from an ancestral diet.
When I was running my restaurant, I made a recipe where I boiled a bunch of pig skin, turned it into literally gelatin, and we had this local black cherry syrup that I flavored it with, and I made this black cherry jello from pig skin, and it was really cool, and literally no one
probably delicious.
Nobody ordered it.
Nobody ordered it,
but no one ordered it, so it was fun.
Well, Jello's gotten a bad rap.
Yeah, for sure.
Well, it sounds like there's a lot of work done, but also many more things that need to be answered. I mean, if you guys could work together to design a system to figure out, or an experiment to test your different hypotheses as to what is the primary cause of obesity related to linoleic acid and seed oils, how might you go about doing it? If you guys could have access to a university and some funds, what would you guys do? Say, hey, let's test our hypothesis.
Well, I mean, you know, a lot of that work's already been done. And the one thing, you know, I think the one thing that really remains to be done is to kind of try and figure out how much can you tweak these systems through different fatty acid. I mean, there's a scientist, John Speakman, who's a world expert on this stuff. And he did a really neat study where he looked at protein versus carbohydrate versus fat and weight gain in rodents. And, you know, what he found was that the signal all comes from fat consumption. And there's a curve. So when you start out at a low fat diet, they don't get obese. When you're in the middle of the fat consumption curve, that's when they get the most obese. And then when you tip down towards the lots and lots of fat and little carbohydrate end, then they start tipping off on obesity, right? And that's the ketogenic diet end of the thing. And he's shown the same thing in another study where he said, you know, it's interesting, the middle range of fat and carbohydrate seems to be the sweet spot for getting overweight. But that said, he only used one fat composition, right? He had a constant ratio of all the fats in the diet. It was just how much of that package of fats was he feeding them. And what really needs to be done, and it's been done by some other researchers, but not quite in the comprehensive manner that Speakman did it, is testing all the different ratios of fats and seeing, you know, so for instance, we have some studies that show that omega-6 fats are protective against obesity. And one of the ways in which they're protective is that they block the endocannabinoid production pathway in the gut, right? So if you take DHA blocks the production of the
Tucker,
hyperphagic endocannabinoids.
I think you said omega-6 there.
Oh, did I?
I think Tucker's talking about omega-3 here. I just want to clarify that for the audience.
If you eat excess omega, thank you. I do this all the time. It drives me bananas. If you eat excess omega, or not excess, but if you eat an appropriate level of omega-3, that it blocks the metabolism of omega-6 into these pro-eating endocannabinoids. And if you look at human populations, we've got the Japanese and the Chinese, who are similar genetic background, except one of them is in the middle of a massive obesity epidemic and the other isn't. And what's the difference between their diet? Well, one of the major ones is the Japanese eat a lot more fish and a lot more omega-3 than the Chinese do. So maybe that's why that's happening, right? But we need to do that sort of research needs to be done to really nail it down and figure out what the best tweak to the system would be, right? Because there are a lot of populations with kind of messed up diets, not because they want it to be messed up, but just because of the economics. So we need figuring out the most economic way of fixing this would be really beneficial.
Yeah, and the other thing I would add to your point about that experiment with the mice is that there are a lot of experiments where we've taken different groups of mice, fed this one starch plus beef tallow, fed this one starch plus soybean oil, fed this one the starch plus olive oil. You can see that the generally, even when the diets are isocaloric and they control the calories, the ones given the polyunsaturated fats become obese. The ones given the saturated fat or the beef tallow typically end up with the least amount of obesity and the least amount of upregulated lipogenesis genes, and they do the best. And so you could imagine in that,
right
now you layer that experiment over the one you were talking about, you've got a really interesting experiment, like you said.
Yeah
because we can also see, right, that the traditional French diet, which is starch and fat,
dairy fat, and animal fat,
right, and it's animal fat, and the French are very lean, or traditionally have been very lean until the past 30 years, as they've added sunflower seed oil that they like, right, for whatever reason.
Well, it's cheap. Same reason we eat it.
Well, yeah. I guess they don't have the soybean processing industry like we do here, so they don't have the.
Well, they've got, Ukraine is the biggest producer of sunflower oil in the world, and they're in the neighborhood. They're the market for that.
Yeah. True.
But yeah, no, that's absolutely right. I mean, that's one of the, the argument that, Speakman's study showed that this middle ground of fat and carbohydrate consumption is the most obesogenic, but we know from human populations that it's not, right? As Brad just said, in populations like the French, where that is their traditional diet, they were traditionally known to be very lean. And that holds for most of Europe. I mean, I haven't been over there in a few years, but generally they're all skinny. They're not as all skinny as they used to be, just like we're not as all skinny as we used to be. But if you look at the American diet back in the 60s, when obesity was a rare thing, that's what we were eating: a lot of animal fat and a lot of carbohydrates.
Yeah, and Americans are still very lean in the 60s, early 70s. I mean,
yep.
See, Tucker, that goes back to the original question, which is, how do you get back there, right? We're all want to know, how do you get back there? And if it's just a genetic thing, well, that would seem to be ruled out by what you just pointed out, that in the 1960s and stuff, people were generally lean, and it wasn't really seemingly isolated to a certain genetic cohort.
Well, it's clearly not genetic, because genetics don't change fast enough to explain our obesity pandemic, right? Because it's happening all around the world. It doesn't matter what your genetic background is. Now, once it happens, then you can start arguing about which genes predispose or protect you from it, which is a nice academic exercise, but it doesn't really help you to solve the problem, in my opinion. Although it can be useful to point out what the problem is. I mean, one of my new favorite papers is looking at a diet called the, of course, it just escaped me, but it's a standard obesogenic diet that pretty much relies mostly on butter. Oh, darn, Brad, I know Sirwit diet, the Sirwit diet.
Right.
So the Sirwit diet is this obesogenic diet that mostly relies on butter and sugar and starch. And they took in Japanese and Chinese populations, showing how genetically similar they are, they have a common genetic mutation called the ALDH 2*2 mutation. And what it is a downregulation of the aldehyde dehydrogenase enzyme, okay? So what they did is they copied this human ALDH enzyme gene into mice, and they discovered that it accelerated their obesity under this Sirwit diet, right? And then they tweaked it. They gave them a stimulator of this enzyme, and then they gave them the thing that the enzyme is supposed to break down. And stimulating the enzyme protected against obesity, giving them the thing that the enzyme is supposed to break down, or I'm sorry, stimulating the enzyme protected them against obesity. And then giving them the thing that the enzyme is supposed to break down is caused obesity, right? And what's that thing? That thing is the molecule that I mentioned before, H&E, which is produced only from omega-6 fats, which are only coming from the diet. So aldehyde dehydrogenase is the enzyme that breaks down, that's part of the detoxification process of alcohol. And it also detoxifies seed oils. And it turns out that in Asian populations, it is associated with not only obesity, but type 2 diabetes and cardiovascular disease and everything else. So a knockout, especially a human knockout of a genetic pathway, can be really helpful in trying to identify what is driving a disease state. And, you know, we have this ALDH 2*2 human knockout, which really points the finger that seed oils are driving the whole thing. You know, and by the way, the real world effect of that, it's kind of interesting. If anybody's ever heard of the Asian flush, where Japanese people who drink alcohol get this red flush on their face, that's because they have this ALDH 2*2 genotype and they're unable to detoxify alcohol. And what's really interesting about that is it's 100% protective against alcoholism. People with that genetic mutation don't become alcoholics because it's so unpleasant.
Right.
But they're also unable to detoxify seed oils.
Right. And so just to clarify, is the genetic mutation where they do not have the gene, which I'm spacing on the name of, but ALDH, there we go. That you just described. So you said it's very predictive in European populations of obesity. You mean the people lacking Asian populations. Sorry, you're right. Asian populations lacking ALDH are more prone to obesity.
That's right. And but what's really interesting about that is that if you look at that diet composition on the face of it, it's a high butter diet. And it would lead you to say, oh, butter's obesogenic. But it turns out that the part of it that is actually obesogenic is the omega-6 fats.
Right, sure. Yeah.
In the butter or something else?
In the butter and in the diet. And that if you stimulate protection against the omega-6 fat, you block the obesogenic effect of that diet.
Would that suggest that if you eat too much butter, you still are getting too much omega-6?
Well, here's one of those areas where, you know, it's mice versus humans. I mean, you know, humans on a high dairy diet don't get obese.
Well, and that's also a case where the diet that we feed the cows has also changed, right? The cows are
that's a very good point.
A lot more omega-6 than they used to. So if you look at the amount of linoleic acid in grass-fed butter, it's a lot less, right?
Right. That's right. That's right.
I think we should wrap it up here, but I want to give you guys a chance to plug anything you guys got going on your blogs or shows or anything that people can look forward to and where they can find it as we wrap up.
Brad, go ahead.
Okay, yeah, sure. So my blog's called fireinaBottle.net. Check it out. There's a lot of nerdy stuff about reductive stress and others, but there's also a lot about food history and the traditional French diet. that we've been talking about. I'm on Twitter at fire_bottle. I'm 90% sure that's my Twitter handle. Those are the two best places to follow along with what I'm doing, as well as Firebrand Meats, where firebrandmeats.com, where I'm making a pigs with a low amount of omega-6 linoleic acid in their fat.
And I'm at
very good pork. Check it out.
I have an easier Twitter handle. It's at Tucker Goodrich, and a harder blog handle. It's yelling-stop.blogspot.com. And I've also got a podcast on YouTube and Libsyn. The Libsyn link is on my blog and at the YouTube podcast. So, Brad, I need to have you on my podcast.
Yeah, let's do it.
We could have a lot of cool conversations.
Yeah, for sure. No, this has been great, guys. I think that was actually really great. Pulling together of a lot of, Tucker and I are obviously involved of a lot of threads that I think kind of start to fit together. So this has been cool.
Well, and I also think that our discussions over things lead people to think that we disagree a lot more than we actually do.
Did you guys learn anything new today?
Yeah, I learned more about reductive stress. I think it's a fascinating area. I mean, the pathway that Brad described of how linoleic acid, causes an acetyl-CoA overflow in the cells is really fascinating.
Yeah, I did as well. I mean, and I have not kept up on the ALDH story, so that was very interesting. So look at this, we're pulling it all together here.
There you go.
Cool. Thanks, guys. I appreciate what you're doing.
All right, thank you.
Thank you, David, for having us on. It was great.
Bye-bye.
Yeah.